We start our lecture, and topic of today's lecture, target therapy. It is lecture number 4. And we need to start from example. It is human on display, on slide, photo of human cell with Gaucher disease. People with this hereditary disease have a defect of gene that codes for glucocerebrosidase. It is an enzyme that breaks down glucocerebrosides and insoluble lipid found in cell membrane. In patients with Gaucher disease, this enzyme cannot function excellent, and this is products glucocerebroside will accumulate in macrophages, macrophages, lysosomes, where it is normally cleaved. Macrophages increased in size, like in this slide, and histologically preparations will visible or look a structure as crumpled paper. Concept, main concept and definition of target therapy. Now, in this slide, you can see DNA sequence, part of DNA sequence, and one node of target. And also you can see bottle with drug, same, this is target. And left part of this picture consists of sum of people, 4 people, and only 2 of them has this target. So if we will use this drug, only part of person can to be sensitive and can have good effect after therapy. It means before therapy always need make genetic testing for treatment, for successful treatment. So target therapy is a therapy directed at tumor target proteins, receptors, genes, attack tumor cells without harming healthy ones. It is also very important because traditional drug therapy, I will say about later, make many bad effects on normal or healthy cells. The principle of targeted therapy is targeted. This is type of targeted is make the goal, genetic goal, selective effect on a tumor cell. Drugs are designed for a specific mutation in a specific gene, like was in our picture. Application of targeted therapy. First, it is a tumor disease. And when I gave comments for this course, cell biology, I said, we will speak about tumor. Example, we'll take tumor disease, but in real, it is transcription factors, ligand, receptor, transcription signaling ways, pathways. It is not only about tumor disease. It is more common. We only will take tumor disease as example, but it will work same and target therapy and signal pathways and transcription factors also work in many disease. Example, autoimmunity disease, especially severe forms such as rheumatoid arthritis, systemic lupus, erythematosis, psoriasis, multiple sclerosis, allergology for treatment of severe forms of bronchial asthma, transplantology for the prevention of transplantation rejection, viral and chronic inflammatory disease, orphan disease, paroxysmal nocturnal hemoglobinuria, cryopyrin-associated periodic syndromes. Common for this group of different disease, genetic mutation, chromosomal mutation, and this is goal for or target for this type of therapy. The appointment of target therapy must necessarily be preceded by a test that allows based on the identification of a molecular predictive factor, receptor, specific mutation, and so on. So identify patients who will benefit from the appointment of this therapy. Need to say that in 27 years, 2 events occurred that changed the usual paradigm of research and use of anti-cancer drug. First, the results of research into the use of immunocheckpoint inhibitors, example pembrolizumab, for the treatment of tumors with microsatellite instability, MCI, have led to a new concept, disease-agnostic therapy, disease-independent therapy. It is a new step of treatment hereditary disease, genetic disease, and will explain about this type on the end of my lecture. But we need return to chemotherapy. It is traditional type of therapy where use common drug, and this type of therapy is associated with the presence of toxicity due to the impact of target that will be common with the tumor in the patient's body, bone marrow cells, mucous membrane. But you can to look for this picture. This is beautiful girl, but he not hair on her head. Head and bone marrow cells. It is cells which, like tumor cells, always divided, and cytostatic effect of drugs make this is chromatin. If you remember, when we was speak about mitosis, we speak during mitosis in prophase, nucleus disappearance, chromosomes materials condensated, and here very excellent target for any chemical drugs and any damaging factors. And chemotherapy, it is action like this is drugs which divide during division prophase, metaphase, make damage of chromatin materials because the DNA will without any safe and very easy can damages. And so all cells, normal cells from bone marrow cells from hair, which always divided, also will very sensitive for this is drug chemotherapy. And so we will treatment tumor, but also will damage all normal cells. How action target therapy? You can to look of these pictures, and you can to look first part of these pictures where you can to see many patients with disease, but they all gray. We not know which mutation their disease consists of. We need make molecular testing or PCR or DNA testing, and then we will to know about prognostic markers or predictors. We will find many type of mutation. Every type of mutation will with different color. Example: yellow, green, blue, orange. It is all different mutations. And then we know that we have many drugs for easy in these pictures, one mutation and as target, and drug for target therapy for action specific for this mutation will same color. So for pink mutation, we need to action pink drug, for blue, orange, and this is pink cannot make action for blue mutation or orange mutation, and so on. What can to use as potential biological targets? About these targets we was speaking in previously lecture. It is receptor, it is ligand, it is primary messenger, second messenger, it is signaling way proteins, and it is transcription factors. Because all these proteins make activity high expression of different genes, and this is genes after mutation will use new action and give oncogenesis. I speak about this in previously lecture, but now I have note your attention what for memory. It is maybe a receptor for epidermal growth factors, e.g. EGFR, and example vascular growth factors, VEGF. It may be proteins that conduct mitogenic signal from receptors. It is molecules that control the launch and course of programmed disease, death, or apoptosis. It is genes for responsible for growth and division. And new, very promising new technology, switching off or silencing. This is technology based on genes and make mechanism off. This is translation or transcription of this is genes. And for this mechanism, we'll use me or small interfering RNAs. This is very interesting new mechanism, and we also will speak about this in our class, practical class, and we will give additional lecture in our research club in department, which will happens in 14th of October. And I invite you in this is additional lecture for you coming and listen. This is additional materials. All these targets are also present in a normal cell, but during tumor transformation, their overexpression or hyperactivation may occur, which is the basis for the use of certain target drugs. Intracellular targets for targeted drug. It will epidermal growth factor, receptor angiogenesis, receptors and proteins with tyrosine kinase activity metagenic, signal proteins directly, genes responsible for growth and proliferation of tumor cells. This is a slide from our previous lecture. You was look this and you know what about this, but I want only give you some example. More wide you can use in next look. This is previous lecture and remember. And also we will speak in our class. Here therapeutic targets for VNT beta-catenin pathway and development therapeutic manners. And here you can look only part. Example, we have ligand VNT and receptors which this is VNT will work. First, A will show VNT soluble receptor. So it will make connection with VNT, and this is VNT after connection with soluble receptor, not will connection with this is receptor, natural receptor, and so not will work. This is complex. Beta-catenin not will free, not will come to nucleus, and not will give cell proliferation. If we'll use freeze-let antibodies, they will make connection with ligand. Now you look on this picture and after 5 minutes, I will continue. I give you some times that you can to think about these inhibitors, and I hope all you was think and now understand. So, Friesland antibody connection with natural receptor. In special, this is VNT work with this Friesland receptor and give free for beta-catenin. But if we have a drug, we can make block first VNT. Second variant, we can block this is Friesland receptor. And in these pictures, we can to see cyclooxygenase, cyclooxygenases inhibitors, which after all work and beta-catenin free, but want to come to nucleus, but this is inhibitors not will give, and so not will come any effect. You need to know there are two main classes of targets drugs. It is small molecules capable of penetrating the cell membrane of interaction with target inside the cells. And in this picture, you can to look example. This is cyclooxygenases inhibitors, which is small molecular. And here, second type or second variant, it is antibody, which second monoclonal antibody or large molecules. They cannot come to inside of cells, and that interaction with receptor or ligands on the surface of a malignant cell or extracellular growth factors. How happens name of small molecules? If drug's name will end in ib, it means indicated that the drug has property of E inhibitors and proteins, ib or ib in Russia because belong, and ib in English proteins. This is small molecule weight drugs less than 900 atomic mass units. The development of the first such drugs dates back on the 19th century. Small molecules still prevail, accounting for about 900 of modern drugs obtained from a chemical manufacturing process, and so in abbreviation or a small name will as a small. Small molecules have the ability to penetrate in the cell and therefore affect not only the target located on the surface or outside the cell, but also the molecules involved in the cascade of intracellular biochemical reactions. Small molecular targets are mainly enzymes and receptors that have activity site for drug binding. At the same time, approximately 7,500 of proteins of the human proteasome do not have such sites at all. For example, in transcription factors, scaffold proteins and not enzymatic proteins, and therefore they cannot fall under the scope of the small molecules. Mutations in genes encoding target proteins can cause conformational change in binding sites that disrupt drug binding. and this is drug resistance. Here you can to look bad effects which cannot give excellent treatment. And position number first, you can to look here many molecules which not have target for connection with drugs. So drugs we use many, but effects not result. Second, off-target effect. Here you can to see two type of proteins. First proteins, it is our target, and second another proteins, but they have same target for drugs. And our drugs can block first our cells and another cells, which function will very need for good function of organism. Situation number 3. Here you can to look that proteins have not one domain, then have 3 parts, and all 3 parts will have any effect for cells. We can block one effect, but second and third will work and not will give effect for treatment. Next, it is reaction when drug will block target on cells, but will work accumulation new proteins, and we will need to give more and more drugs, and effect not will come. And next, about after mutation, when was target round, but after mutation will with a square, and our drug cannot connection and so not have give any effect. If example, I need to say you about Janus kinase state signaling pathway. This is work and make separative state which will translocation in nucleus and give expression effect for target genes. This is drug for make this is drug upadacitinib, ending ib, will give inhibition effect for Janus kinase and need for make this drug $2.5 billion. It is very high price. But here will block this separative state, and so not will work in nucleus and not will give any signaling effect. Next, voxia-leto. Need for make these drugs near $1.8 billion. And here in this is picture you can to look normal erythrocytes and erythrocyte with mutation. We speak about this in our medical biology class. They will have mutation in hemoglobin LEDs and give new shape, which not excellent. But this is drug will binds to hemoglobin, inhibits its polymerization, and increase connection hemoglobin with oxygen. It will prevent red blood cells from deformation and restores normal oxygen delivery to organs and tissue. And next, it is Zolgensma. For this need to take $1.6 billion, and this is construction vectors which will building with adeno-associated viruses vector, which will have normal gene. Disease for which use this drug, it is atrophy of nervous tissue and small baby cannot speak, cannot move, cannot walking, cannot sitting, and in end of their life they cannot make breathing and die. If this is construction will use for treatment, this is gene with viruses will come to DNA in every nervous cells and make new healthy variant, and all nervous tissue will divide it, cells will divide it, will make new normal tissue, neurological tissue, and all will excellent. Next, it is how will happens names of monoclonal antibodies. It is end in MAB. Monoclonal antibody, M monoclonal, anti, A from anti, and B from body. Also, it may be have more extended CMAB. This is ending speak to us or explain to us for a chimeric antibodies approaching human antibodies, however, contain part of the mouse proteins. I will explain next how it happens. And maybe MUMAB for refer to fully human antibodies. And look of these pictures. It is antibody structure. It is antibody which we have. And this is protein immunoglobulin with a given targeted specificity for a particular antigen. This is information give antigen presented cells for B cells, for T cells, and they will foundation and generation antibody with special antigen information about viruses or about any gene. Then this is B cells, B lymphocyte can make clone of cells and foundation many monoclonal antibody which will all identical and give make this is information. The first commercial monoclonal antibody, orthoclone OKT3 or muronomab CD3, was approved by the USA Food and Drug Administration. It is organization which give permission for all drugs which will use in USA and in the world. In 1986, for the prevention of kidney transplantation rejection. Completely murine antibodies therefore had a high immunogenicity, and so first want to win transplantation damage from immune cells, but in result this is drug will also make activity immunity and also give antibody, human antibody against mouse antibody which was used in drug construction. Here you can to look types and how which make these drugs. The first antibody give to mouse. It will immunize of mouse, and they will make B cells with B cell will with many antibody. But more good for this is function will use tumor cells, myeloma cells. It is tumor from B cells, and you remember all tumor make many division, and if make this is hybridoma, combine these properties and this structure, it will hybridoma cells that produce it monoclonal antibody. But poor mouse antibodies are rejection by the human immune systems. So need make new genetic research and use genetic engineering manipulation for make more good. In one of technology, this is will active epitope from mouse antibody with human immunological proteins, and this is hybridoma will make more natural for human organism, and immunogenicity, activation of immune response will down. And here you can to see chimeric variants of chimeric monoclonal antibodies, which was used in future research. And chimeric we will speak when we will cross mouse and human. And if it will big part of mouse and more part of human, it will chimeric combine. But if 90-95% of human immunology and only small part from mouse, it will humanized antibody. In the 30-year science of this research was when was got first drug, uh, development of therapeutic monoclonal antibodies production, and it has evolution from mouse to human, and development many new targets, and have many more good for human organism technology. The total number of drug, it is one stage or another of development in the world, was more than 570 was registered at 2018. The introduction of drugs of this class has made it possible to reach a new level in the treatment of a wide range of interactable disease. And here you can to look, this is evolution. This is scheme of structure mouse, chimeric, humanized, and human antibody. Here full mouse proteins, and we speak about mouse antibody hybridoma, which was changed with myeloma cells. This is only 34% of mouse proteins, and this is chimeric antibody. This is only 10% of mouse proteins, so this is humanized antibody. And here full human proteins, so it is recombinant human antibody. And this is scheme to give you possibility to be specialist or expert, and you can in name of drugs to understanding how will work immune active responsible, what it is type of antibody, mouse, chimeric, humanized or human, and how many part of DNA sequence will include mouse proteins and will include human proteins. And this is nomenclatural name: MOMAB, full mouse; XIMAB, more part of human; chimeric antibody; ZUMAB, more 90 part of human, humanized; and UMAB, full of human proteins. And now I will need to give you example. It is tumor site, and it is tumor site only one part of glandula, mammillary glandula. And here metastase, it was come from blood and lymph way, and maybe this is will one gene mutation may be different. Need to make first DNA PCR research. In research, we will find that we'll have many receptors. This is cytoplasm, this is cells inside, this is outside. This is outside part of receptor HER. Maybe different type, but only one type will special for oncogenesis and not will give any effect for treatment. So we need to make new drug for block this is second type. And this is drug will trastuzumab, and from this is name and from this is end you can to understand which type this is drug. It is humanized antibody and 510 DNA from mouse, 95% from human. And this is picture show how it work. This is cells of tumor and have many receptor H, E, R, different type. And here our drug. And they will come for special second type of receptors and inhibition. Inhibition, this is effect and not give any result for divided of tumor cells. Second slide, this slide will give to you same many different another type of drugs, ipilimumab, atezolizumab, nivolumab, which work with another type, different type of receptor and give many different effect for tumor cells, which will make block. And also one example, bevacizumab. This is also recombinant humanized monoclonal antibody. Now you understand what it means. And this is will action as against vascular endothelial growth factor, VEGF. And in normal situation, this is VEGF will connection with special receptor and will give any primaries and secondary messenger way and give any transcription of genes. But this is anti-age drug will make block and not will give to action with receptor and not will give any results for gene transcription. And also you can to look here example which you know from medical biology. It is translocation part of 9 chromosomes and connection with part of 22 chromosomes. It is Philadelphia chromosomes. This is mutation will lead to chronic myeloid cells leukemia. And you can to look here, and previously I told you about this. Promoter part will connection in this is chimeric gene with regular structure of ABL gene and will give information for work and new type, and it will give many effects for cell division, for block apoptosis and will leukemia. But drug imatinib will make block this is tyrosine kinase, which will work after activation of this is expression gene, and this is will reduce blood cell lifetime or apoptosis will high. Blood cells will do not accumulate in bone marrow, and blood cells will divide it and will make differentiation and will make to neutrophil, basophil, eosinophil, and will make normal hemopoiesis. And also you can to look about imatinib also, and here how it will work in chronic myeloid leukemia. And here you can to see this is enzyme which will foundation after this is chimeric gene, and for their work need make complex with RTF 3-phosphate macroerges connection, and they will no, after this activation tyrosine. This is tyrosine will make bad effect for cells and give leukemia, oncogenesis. But if we will use, this is our target imatinib drug, they will make place besides ATP, and tyrosine cannot be activated. So effect not will be, and disease will reduce. This is also you can to look another effect of imatinib, and we will speak about this in our class. Our teacher will help you make this is pathway, how will work if will make gene mutation, and how it will reduce if make target therapy. And in last our class you will to know mutation and to have information about target therapy, give for this is structure, for this is patients target therapy, and discussion which gene will work or not will work, and which effect on cells will in result. Next example, it is rituximab. It is CD20 receptor inhibitor. And here you can to look how it works and which effect will give for treatment. Also example about monoclonal antibody and how it will work. Also will inhibition, what this is CD20 receptors, and not will give to work. And here you can to look the third type of drug therapy, target therapy. I said in start of lecture about this it is advanced therapy. How it work? You need to remember about this info. Here we will use genetic construction. And example, we know about one cells present mutation in gene number A. And we need to make drug which will lead to this cells and will make outside mutation gene and inside and ligase with normal gene and it will give effect for successful treatment. What need to use? Need to use adeno-associated virus in which we will make new normal genes. Then as all viruses, they will come to cells and will make restriction, deleted this is mutation genes, and make new construction. And here you can to look that these viruses come to the cells and give new special normal information, and it will happy for all doctors, cells and patients. And I want to say that in cell biology we will speak about this is target therapy, but who will change immunobiology elective discipline, new discipline, which will in your second spring semester you will also to know how make this is, how make vector construction, how make genetic cloning, and we'll try make it in your practical class also. And here, no, some info from lecture. This is lecture you can to find in PPT lecture, which I will give you or make photo, and will find this is lecture about CRISPR-Cas9 technology. This is research woman. It is based of this is technology, and they in this lecture speak about bacterial immunity, which was formed by evolution mechanism and has defensive system of microorganisms that protect them from the consequence of infection by bacteriophages. The principle of this protection is based on the accumulation in the bacterial genome of DNA sequence characteristic of bacteriophages arranged in the form on inserts, spacers, this is part, separated but repeating nucleotide sequences. When a bacteriophage enters in bacterial cells, the complex of Cas9 and RNA read from CRISPR's bind to its DNA and regions complementary spacers. Recognizing such region, Caspase 3 cuts them out, leading to the death of the viruses. And this is woman speak about this is technology can use for treatment. This is adeno-associated viruses also because this is bacteria can from this is Casper's technology will make division, extradition, bad mutation genes, restriction, then make new normal gene led sequence, DNA sequence, ligase, and it will work very excellent. What here you can to look donor DNA, which will construction in old DNA sequence and has new information. And as some more example, you can to look of this is slide, and here information about inhibitors of angiogenesis, avastin, which block many effectors which signaling way and can use in colon cancer, non-small cell lung cancer, cancer mammillary, and cancer kidney. Also, more information about target therapy. This is temozolomide, everolimus, vermouthvenib, trametinib, and dabrafenib. All this is example we will speak in our class, and our teacher will help you. And here you can to look example. More example I will give you in class. This is example I'm afraid in Russian language. And it was finish of our lecture, and you need come to class and we will work with this topic. Thank you for attention and we will meet later.